so this macrophage, just a quick review, he ate something, he blew it up, took a piece of what he ate, and he displayed, remember we call that an epitope. So he displayed an epitope on his MHC class 2. So now we have to think, okay, what comes next? We're going to have something in the third line of defense come, right? What attaches to MHC class 2? If you look back up here again, MHC class 2 always attaches to CD4. And where is CD4 found again? Helper T cells. So in response to the MHC class 2, we'll have a helper T cell come with his CD4 receptor. Now, you have to kind of think big picture here. What is the whole point of this process I'm drawing out? To get immunity to this pathogen, to this microbe that the phagocyte ingested. Now, do T cells get immunity? Well, yeah, they'll turn into memory cells, but the big thing here is developing antibodies. Do helper T cells make antibodies? No. What makes the antibodies? Are the B cells. B cells are inhumoral. So we still have a couple more steps, right? We want to get B cells up here, but we're only stuck at helper T cells, which is great because we have our MHC class 2, which presents the epitope to CD4 positive receptors on helper T cells. The helper T cell, what is his job again? To help, to see, did you eat something really, really bad, or did you just eat dirt? So the helper T cell attaches to the MHC class 2, and he analyzes the situation. He says, whoa, you ate tuberculosis, or you ate the Black Plague. And so now we need to make antibodies against that. So in response to this interaction, if it's something bad, the helper T cell will release cytokines. Remember, cytokines are chemicals that our cells use to talk to each other. So he releases these cytokines, and one, it tells these phagocytes, hey, there's something really bad in the area. You need to keep eating in order to clear this microbe from our system. Now, keep in mind, he's not telling the macrophage, keep eating that microbe, because the macrophage, or the dendritic cell, or the neutrophil is just some kind of phagocyte. He's in the second line of defense. He doesn't have memory. He doesn't remember what he eats. So this helper T cell is just telling the phagocyte, keep eating, don't be lazy, just eat things. The biggest thing that this helper T cell does when he releases cytokines is recruit B cells to the area. Remember, B cells are what produce the antibodies, so we need to get B cells in the picture. So the cytokines are released, and that tells B cells to come to the area. The T cells are like, hey, we're going to release cytokines, B cells come, we need your help right now. So the B cells come in response to the cytokines. But how do they talk? Well, we do know one thing, right? Helper T cells always, always, always have CD4 positive receptors. And what attaches to CD4 again? MHC class 2. But wait, I thought only phagocytes have MHC class 2. No, if you remember, on this board, I said that MHC class 2 can be done by phagocytes and B cells. That's why it's so important that B cells do MHC class 2. B cells do MHC class 2 so that they can attach to the CD4 receptor found on the T cell. If B cells didn't have MHC class 2, they couldn't talk to the helper T cell because CD4 always attaches to MHC class 2. So that's important. Remember, B cells, we always tend to focus on the phagocytes being able to do MHC class 2, but phagocytes and B cells do, and that's important in this part. So what's being said right here? The helper T cell is telling the B cell, hey, I was talking to this macrophage today and he ate something really, really bad. You need to make antibodies against it. Now that this B cell is activated, since the B cells are lymphocytes, they're in the third line of defense, they're going to make memory cells. So it's going to make memory B cells. And that's really important for the second time you get exposed. Because the second time you get exposed, you don't have to go through this process, you just skip right to the memory B cells. Which is why you don't get sick, because this process takes a really long time. But more importantly, for the first exposure, this B cell will differentiate into plasma cells. Plasma cells are important because they are the cells that secrete antibodies. Remember, we're talking about the first time that this person has been exposed to this pathogen, and the first time that person is exposed to a disease, the B cells secrete IgM. That's the first exposure. Now, if we want to make our picture complete, we would also say that this helper T cell will also create memory T cells. Because remember, these are lymphocytes, so they do create memory cells. So let's look at the big picture here. We need to make a couple points. From here down, if you look closely, we have a phagocyte. Remember, it could be a neutrophil, macrophage, or dendritic cell. This is the second line of defense. From here up, what do we see? T cells and B cells. So all from here up is the third line of defense. Remember how I've been telling you this whole time that a lot of people think, oh, the first line defense, and when it fails, it goes to the second line defense, and when it fails, it goes to the third? That's not right. They all work at the same time. A perfect example is right here. The second line of defense actually works with the third line of defense. Even more than that, everything in green, we see T cells, right? And what branch of the third line of defense are T cells in? They're in the cell mediated. So all of this green is cell mediated. Okay? But last time I checked, B cells and antibodies aren't in cell mediated. What are they in again? They're in humoral. So up here, we see B cells and antibodies. So this is humoral. Remember, cell mediated and humoral are both in the third line of defense. They're just different branches. Helper T cells, cytotoxic T cells, any kind of T cell is cell mediated, whereas humoral are B cells and antibodies. So if you look at this carefully, the only reason humoral happens is because of the helper T cell. Helper T cells, we like to say, are the bridge between cell mediated and humoral. They're very, very important. If you look at it, macrophages can't talk to B cells, because macrophages have MHC class 2, B cells have MHC class 2, so that doesn't work. That's why the helper T cell comes in. He helps. That's good for now. Let's move on to the other picture, and then we'll compare the two. So, not my house right now, so I don't have very much board space, but I'll make it fit. Hopefully you guys can read this. Alright, in this example, we're just going to start right off the bat. We're just going to start with maybe like an epithelial cell, a cell in my finger, okay? And let's say that this cell, that's just a nucleus, by the way, because we're saying it's just one of your cells. Let's say he's infected with a virus. Okay? Now, remember, we're just talking about a cell in your finger. So, we need that cell to do some kind of MHC. So let's look back at our board here. We're not talking about a T cell right here, so we don't even focus on the CD4 and CD8. We're just going to look down here at the MHCs. So we have the choice between MHC class 1 and MHC class 2. Okay, our first question is, what kind of cell are we looking at? We're looking at a cell in my finger. So, any cell infected? Yeah, a cell in your finger can fit into that category. MHC class 2 is only B cells and phagocytes. That doesn't include the cells in my finger, so that's off. The other thing that makes me pick MHC class 1 is the keyword infected. I said that this cell was infected with a virus. Even if it was a phagocyte, remember I said phagocytes can be infected with the virus as well. So if I drew a phagocyte infected with tuberculosis right there, it would do MHC class 1. If I drew a helper T cell, it could also do MHC class 1 if it's infected with HIV. I could draw a B cell, and it could also do MHC class 1. Remember, MHC class 2 is only B cells and phagocytes, whereas MHC class 1 is any infected cell. I could draw a B cell there, I could draw a T cell there, I could even draw a cytotoxic T cell there if I wanted to. The key is it's infected. But I'm just going to use the typical example of just a cell in your finger. I hope that made sense. Any cell that's infected can do MHC class 1.